Hello friends, today we are discussing about metagenesis in Obelia. What is metagenesis? It is true alternation of generations occurs where a diploid or sexually reproducing generation alternate with a sexual haploid generation. For example, metagenesis in sown Obelia in a Cnidarian. So a brief introduction about it. Commonly known as C4, Obelia is sedentary marine colonial form found attached on the surface of seaweeds, molluscan shells, rocks, and wooden piles in shallow water. Each colony of Obelia consists of horizontal thread-like root called hydrorhiza which is attached to a weed or any substratum. From hydrorhiza arises a vertical branching stem about 2.5 long, which is known as hydrocolus. The hydrorhiza and hydrocolus are hollow tubes. The hydrocolus bears zoetes or polyps on either side in cymose formation. Each polyp has a stem and terminal head called hydranth. The hydranths are feeding polyps. They feed by capturing minute animals and larvae. Towards the base of the hydrocolus, in the axis of the polyps, are reproducing polyps called blastostyles. The entire colony is covered by a tough yellow chitin secreted by ectoderm. This covering is known as perisac. The perisac around a blastostyle is a gonotheca. The blastostyle and gonotheca are together called gonangium. The Obelia is a trimorphic colony that is having three kinds of zoetes which are as follows. Polyps or hydranths, that is nutritive zoetes, then gonangia or blastostyles, that is budding zoetes, then medusae, that is sexual zoetes. Life cycle of Obelia. Life history of Obelia includes both sexual and asexual generations that alternate with each other to complete the life cycle. The germ cells originate in the ectoderm of the manubrium quite early when the medusa itself remains attached to the blastostyle. When the gonads are ripe, ectodermal covering ruptures and the germ cells are set in water. The ova are large rounded cells. The sperms are minute, actively swimming flagellated cells. The medusae die soon after liberating the gametes. Then fertilization occurs either in the seawater where the germ cells are set free, or spermatozoa may occur by water currents to the female medusae or fertilize the ova in situ. Zygote formed after fertilization immediately undergoes cleavage. The cleavage is holoblastic and a blastula is formed. By evagination, the blastula is converted into an oval ciliated planula larva. The planula consists of an outer layer of ciliated ectoderm and inner mass of endoderm cells enclosing a space, the rudiment of colenteron. The planula swims freely for a brief period and settles down on some submerged substratum by one end. The proximal end gradually narrows down and disc appears for attachment. The distal end expands and by developing a manubrium and a circlet of tentacles it turns to be hydrula or simple polyp. The hydrula sends out lateral buds and by repetition of this process it is converted into a complex Obelia colony. Alternation of generation of Obelia. A distinct alternation of generation or metagenesis is present in the life history of Obelia. The Obelia colony is sexless, bears no gonads, and develops by asexual process, that is by repeated budding of the hydrula. For the medusae birds, some of the zoetes of the colony develop gonads and from there fertilize egg, new Obelia colony arises. The asexual generation is dependent on and is alternated by sexual generation. Obelia is permanently fixed colony, but the Planula larvae it produces are free swimming, thus a non-locomotory species becomes locomotory.